Okay, so welcome to this video. We'll be looking at uh, questions from test 2, and this test 2 was on biochemistry. Okay, so we're expecting to find questions on proteins, carbohydrates, lipids, nucleic acids, okay, all those things, enzymes, all the biomolecules. We're expecting to find questions in this paper. So in the first session, okay, we started revising this paper. So in the first video, we actually did question 1 all the way up to question 32. Okay, so if you didn't see that video, it's there on the website. Okay, just go in the past papers, you're going to find it. So in that first video we did from this question 1 that you can see here. So from question 1 all the way up to 32. So all the way up to here, we solved all those questions. So you can refer to that video. It's there on the website or on our mobile app. Okay, and then for today, we're going to start from question 3 there. 33, I mean. We're going to start from question 33. So let's start. Okay, so question 33 reads, what is the meaning of a solid wedge? Okay, so when you talk about stereochemical drawings, okay, there's a notation that helps us to visualize 3D structures. Okay, so for example, if I have a carbon here, and then you have a solid wedge bonded maybe to OH, and then somewhere here it can have a dashed wedge like this, bonded to hydrogen like this. So this one here should be should be shaded. Okay, so this one here, the wedge. Okay, both this guy and this guy are called wedges. Okay, but this one here, which is shaded, this one here, it's called a solid wedge. Okay, this is a solid wedge, and then this one here is a dashed wedge. Okay, dashed wedge. So both of these have different meanings. Okay, so when we're talking about a solid wedge, okay, when we're talking about a solid wedge, then this solid wedge will be representing a bond that comes out of the plane. Okay, that comes out of the plane of the paper towards the observer. So if you are looking at this structure, okay, the bond that will be pointing towards you, that's the one that you represent by a solid wedge. Okay, and then for the bonds that are going away from you, okay, they will be represented by the what? The dashed wedge. So here, the best answer that we can pick will be A, because the solid wedge represents a bond pointing towards the observer. Okay, and then the dashed wedge here, in case it comes in your other test, okay, it will be a bond that is projecting away from the observer. Okay, and then what do we call this thing that is on C here? They're saying a bond in the same plane as the chiral center. So a bond in the same plane as the chiral center is just called a plane line. So for example, in those bonds that we did in organic chemistry, where we had such things here. So you can see this is just a plane line like that, right? Okay, so this one, it represents a bond in the same plane as this chiral center. Okay. Okay, so the correct option here, we're going to pick A. And I've explained this option D here was just a nonsense option to just confuse you because they wrote James Bond. So James Bond, what has James Bond done? Okay. All right. And then 34, they're saying which of the following amino acid is not ketogenic? Okay. So we know that amino acids are classified into 3 groups. So the first one is the ketogenic group. And then the second one, okay, will be the glucogenic. Okay, so this is G-glucogenic. So glucogenic group, this is the second one. And then the other one, it is both, okay? Both ketogenic and glucogenic. Okay, so these are the 3 classifications of these amino acids. So what does it mean to be ketogenic? Okay, so ketogenic simply means the amino acid produces acetyl-CoA. Okay, and then glucogenic simply means the amino acid will produce intermediates, okay? Or it will produce something that will go on and enter glucogenesis, okay? So that's what it means, glucogenic here. So of these amino acids that we've been given here, leucine and lysine, these ones here are purely, okay, are purely ketogenic. So these ones, we are going to classify them as ketogenic what? Amino acids. And then histidine, it is well known that it's glucogenic. Why? Because histidine Okay, histidine forms what we call as the alpha keto glutarate, I mean. Okay, it forms what we form the alpha keto glutarate. So this alpha keto glutarate, okay, it goes into the glucogenesis process. So this histidine here is a glucogenic amino acid and it's not ketogenic. So the option that we're going to pick here would be option C. Okay, so option C, that's the best answer there. Okay, what about question 35? So here they're saying at what level of structure organization would a protein be functional? Okay, so a protein is functional at the tertiary level. Okay, so why is it functional at the tertiary level? Because that at the tertiary level, that's where you obtain a 3D folding. Okay, a 3D folding of all the single polypeptide chains. Okay, so this 3D folding, okay, it will determine one, the protein's shape, and it also determine the protein's function. Okay, so if a protein is not yet in 3D, then it won't be functional, it can't work. So that's the reason why option A here is wrong. Because when you talk about the primary structure or the primary level of a protein, this is just a sequence of amino acids, right, that are joined together. Okay, and then secondary level, um, the bonds that include the alpha helices, the beta pleated sheets, but they are not yet full H3D. I mean, they're not yet through 3D, so they won't be function. So the protein is only functional when it reaches at what? The tertiary level. So the answer there is C. And then we can also talk about quaternary. Okay, so quaternary only applies to multi subunit proteins. A good example is hemoglobin. Okay, hemoglobin has multi subunit proteins. So um, for a protein to reach quaternary, it means it has already passed tertiary. So it will also be function at what? At quaternary. Okay. So sometimes this question they ask it at which two levels of protein organization, okay, is the protein functional? So you choose the tertiary and the quaternary. But when they just say one level, okay, then you have to go for the tertiary. So the answer there is C, which is tertiary. Okay. And then here they're saying an amino acid with two asymmetric carbon is, okay? So an amino acid with two asymmetric carbon is well known to be um, threonine, okay, threonine. Okay, so this one here, threonine has two asymmetric carbons. So what is an asymmetric carbon? An asymmetric carbon is simply a chiral center, okay, meaning a carbon that is attached to four different groups, okay? A carbon that is attached to four different groups. So another good example that has two asymmetric carbons is isoleucine, okay? So if there's no this option, just know that isoleucine also has two. The rest have one, and then this glycine here has zero. Why? Because glycine, it will have a carbon here, okay? And then it will have H there, it will have H there, and then here it will have COOH, and then here it will have Na2, like this, okay? So because this glycine is attached to two, okay, is attached to two hydrogens like this carbon here is not what? Asymmetric. So it has zero asymmetric carbons. Alanine has one, serine has one, and then threonine has two as well. And then another example that you can consider there is also what? Isoleucine. So isoleucine also has two asymmetric carbons. So we choose D as the correct answer there. Okay, so let's look at the next question. Okay, so the next questions will be based on this sequence of amino acids that we've been given. Okay, to be based on this sequence of amino acids that we've been given. Okay, so let's continue. So I was saying the next questions will be based on this sequence of my amino acid, which is being given in the word electricity. Okay, so they're saying a peptide has a sequence, okay, electricity like that. So each of these letters represents an amino acid, okay? So maybe I should mention what they represent before we do anything. So E represents the amino acid glutamate, Okay, L is for leucine, E is again glutamate, C is for cysteine. Okay, and then T is for um, threonine. Okay, and then R is arginine or arginine. I is isoleucine. Okay, C again is cysteine, I isoleucine, T uh, threonine. Okay, and then Y represents uh, tyrosine. Okay, Y represents tyrosine. Okay, so these are the amino acids that are in this sequence. Okay, so they're saying now that note that this sequence represents one letter abbreviation of amino acid in a peptide. Okay, use the given sequence, the given sequence to answer 37 to 42. So let's start with 37. So 37, they're saying what amino acid, okay, is at the N terminal. Okay, so we know that in each amino acid or in each peptide, there's the N terminal and there's the what? The C terminal. Okay, so the way this has been given here is electricity. Okay, the amino acid at the end here is the one that is at the N terminal because that's where you are going to find the amino group. And then the amino acid that is at the far right there, that's where you're going to find the C terminal because that's where the carboxyl group will be. So E represents glutamate. Okay, so the amino acid that is at the N terminal is glutamate. So you pick C. Okay, and then we go to 38. What amino acid is the C terminal? So the C terminal is this guy here, which is Y. Okay, so meaning that you have to memorize, guys. You have to memorize what each letter, okay, stands for in terms of amino acids. So stands for um Y stands for tyrosine. Okay, Y stands for tyrosine. So we're going to say the one that is at the C terminal, this Y here stands for tyrosine, just be C, tyrosine. Okay, and then how many amino acids in this sequence, okay, have um how many amino acids in this sequence have a non-polar side chain? Okay, so this one goes back to the classifications that we did, okay, in my first video that I taught about amino acids. So we said they can be classified also as non-polar, polar, charged negatively, charged positively, okay? So from that discussion, okay, we said the ones that are non-polar, okay, the ones that are non-polar were these here, okay, were these here. So we can see that from this, the non-polar ones, we have leucine, this guy here. What is happening to my pens? It's not working, eh? Okay, I know why. Okay. So the non-polar ones, you can see we have glycine, alanine, protein, valine, leucine, isoleucine, methionine. Okay. So on those options that we've been given, we're just looking for an option that has these amino acids. If we can't see, if we have an option where there are some amino acids that are not here, then that will not be non-polar. So here they're saying how many amino acids in this sequence have non-polar side chains. Okay, non-polar side chains. So we start with E. E is glutamate. Okay, so glutamate is actually polar. So I'll just be writing P if it's polar. So glutamate is polar. And then leucine, we saw it on this list here. It's there. So this is non-polar. Okay, so leucine is non-polar. I'll be putting ka N here. What is happening now? We're putting ka N for non-polar. E, it's polar. And then this C is cysteine. Cysteine is polar. Threonine is polar as well. And then R represents arginine. Arginine is also polar. Okay, I represent isoleucine. So isoleucine, I see it here, which is non-polar. So I'll put there N for non-polar on I. C, we've already said it's polar here. This I is non-polar. P represents threonine, which is polar. Okay, and then Y represents tyrosine, which is also polar. So how many non-polars can we see? I can see 1, 2, 3. So I'm going to pick 3 there. Okay, so it's very important to memorize. Okay, one, the abbreviations. Okay, first you have to memorize the 3-letter abbreviations for these amino acids. You also memorize the 1-letter abbreviations, and then you memorize their groupings based on different stuffs. So you can see like some of the questions that we solved above there, we are uh, we are grouping these amino acids into ketogenic, glucogenic. Okay. You also have to know their distinctions in terms of their charges. Okay. Are they polar? Are they non-polar? You also have to know. So they are the best answer is just 3. Okay. And then 4. Okay. 4, what are they saying there? So 4, they're saying, I mean 40, not 4. At 4 now. 40, they're saying which 2 amino acids contain the hydroxyl in the side chain? Okay, so which 2 amino acids contain the hydroxyl in the side chain? Okay, so the 2 that contain hydroxyl in the side chain, okay, is just uh, thionine and tyrosine. Okay, so if I can show you the pictures here, I don't have tyrosine here. I don't have tyrosine or thionine. Okay, so tyrosine is here. So tyrosine contains, okay, tyrosine contains the hydroxyl uh, side chain. Okay, you can see it there. And then the other culprit is uh, thionine. Okay, so thionine, where are you? Okay, so thionine is here. You've seen this thionine. So you can see the OH group here. That is the hydroxyl group. So you have to memorize. So thionine, okay, contains the OH. Okay, and tyrosine here contains the OH. Okay, so make sure you memorize those 2. So they contain the OH, so are the ones that we're going to pick. So we know that thionine is represented by T, okay, and then tyrosine is represented by Y. So it means we'll go for option D there. Okay, this option here, because thionine is T and then tyrosine is Y. Okay, so those are the answers for part 37 to 40. We move up, we go to 41 and 42. So how many peptide bonds would be needed to join these amino acids? So these amino acids are electricity, like this. So let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So if there are 11 amino acids, then it means we have how many peptide bonds? We have 10. So the general formula is that if I have n amino acids, okay, if I have n amino acids, then the number of peptide bonds will be n minus 1. Okay, so you can see I have 11 amino acids. The number of peptide bonds is just 10. So pick B as the correct answer. Okay, 42. Amongst the listed amino acids in this sequence, which one has the bulkiest or largest side chain? Okay, so the one that has the bulkiest or the largest, this one you should know by heart, of the amino acids, okay, it's tyrosine. Okay, so tyrosine has the largest uh, side chain or the bulkiest side chain. Why? Because in each side chain you find a benzene ring with a hydroxyl group. So they form what we call as the phenolic ring. Okay, so another amino acid that has a large side chain is adenine. So adenine is long, but it's not flexible. So because thyroxine has the aromatic ring, it makes it more bulky and rigid. So let me show you the pictures here. So we're talking about tyrosine, right? So tyrosine is here. So look at this tyrosine here, this guy, the tyrosine there. And then I mentioned that the other one was adenine. So the adenine is here. Okay. So you can see this adenine, you can see it has a lot of tumor bond. This is the side chain. Okay. And when you compare it, you may say that this one is bigger, but no, it's not. Okay. So adenine is long. You can see it's long like this, but it's flexible because these are just carbon to carbon, N to carbon bond. So it can bend in either way that it wants. Okay. But this one here, because it has this ring here that is connected to OH, it forms what we call as a phenolic ring. So the phenolic ring is rigid. It's not easy to break. Okay. So it's rigid and it is bulky. So it makes this tyrosine to have the bulkiest side chain. Okay, so the one with the bulkiest side chain is tyrosine. Okay, followed by arginine. And then the one with oh, what am I not? The one with the lightest, okay, or the smallest side chain is just glycine. So for glycine, you can see we just have like a hydrogen there. That's the side chain. Okay, so it's the one that has uh the smallest. So in case they ask you that, you know what to answer. So here I'm just going to pick tyrosine. Okay, so of the given amino acid, tyrosine is the one that has the bulkiest side chain. Okay, so we are done with that question on electricity. We go to question 43. So 43, what are they saying? Okay, so 43, they're saying, which of the following is not an example of a protein involved in immunity? So here, we are looking for something that is not a protein. So cytokines, these are proteins. Antibodies are proteins. The MHC proteins, they have already said proteins. So these are proteins. So the one that is not a protein here is this one, the white blood cells. These are cells, they are not proteins. Okay, so we choose that. Um, there. Okay, so 44, there are only three amino sugars. Okay, which of the following is not one of the amino sugars? So the amino sugar that are there is one, glucosamine. Okay, glucosamine. This one here, it's an amino sugar. Galactosamine, it's an amino sugar. Manosamine, this one here is also an amino sugar. So these ones here are the three, okay, amino sugars. And then this fructosamine here, this one is not an amino sugar, okay? So fructosamine is not an amino sugar. It's a glycan. It's a this word, I forgot to pronounce this word. It's a glycation product. Okay, it's a glycation product. That's what fructosamine is. So when you get fructose and then you add an amino compound, okay, uh, that's when you form fructosamine. Okay, so it is found in proteins like glycated hemoglobin, okay, but it's not a natural amino sugar. So we only have three amino sugars, so you should memorize them. We have glucosamine, galactosamine, and manosamine. Okay, those are the amino sugars that we have, three of them. Okay, let's look at 45. 45, they're saying when an aldehyde or a ketone, okay, is in equilibrium with an eno group, okay, or just say an eno, due to the transfer of electrons and a protein, the process is called what? Okay, so this question is direct from the tutorial sheet on the carbohydrate. You can check it out. Okay, so the answer here is tautomerism. So this tautomerism, that's a situation where there's a reversible structural isomerism, okay, involving the relocation of a hydrogen atom and the what? And the double bond. Okay, so you can move from an aldehyde group or from a ketone group to an eno group, okay, using what we call as the tautomerism. Okay, so that's the correct answer there. And then this mutarotation is not correct. Okay, so what is mutarotation? Mutarotation is the change, okay, between the alpha um, and the beta anomers. Okay, and then love triangles, this is just a nonsense option. It's not supposed to be there. And then stereoisomerism, this is just the general category. Okay, it's not specific to the keto-eno shifts. Okay, so the best answer there is tautomerism. Okay, 46, they're saying which polysaccharide is used clinically to measure the glomerular filtration rate. Okay, so the one that is used to measure the glomerular filtration rate is inulin. So this one is a common question. You may uh, just have it at the back of your mind. Okay, so the polysaccharide that is used clinically to measure glomerular filtration rate is inulin. But why is inulin used? Why not peptidoglycan? Why not heparin? Why not glycogen? Okay, so inulin is a fructose it's a fructose polysaccharide that is freely filtered at the glomerulus. Okay, so since it's freely filtered at the glomerulus, okay, it is used, okay, to measure what we call as the clearance. So the clearance equals to what we call as the glomerular molar filtration rate. Okay, so because you have these tubes that are coming here in the glomerulus and then they are going there. So you have a flow, okay, of this urine that is coming there. Okay, so if you add inulin, okay, or you inject inulin into the patient's blood, when it comes here, it won't be absorbed by this glomerulus. Okay, instead it will just pass through like this. So since it is freely, okay, since it is freely filtered at the glomerulus here, we can use it to find the glomerular what? Filtration rate. Okay, but if you put something that is absorbed here, then we can't know. Okay, because some of it might be absorbed here and then some of it has come out. So we can't really measure the the glomerular filtration rate like that. So we need something that is free filtered here so that all of it goes this side. Then we can measure this rate here. Okay, and then what is peptoglycan? This peptoglycan is just the bacterial cell wall polymer. Okay, so it is used or it is found in the cell wall for the bacteria. And then heparin, okay, heparin is an anticoagulant. So meaning it is used to prevent the blood from clotting. Okay, and then glycogen, that's just the storage carbohydrate. Okay, it's not used to measure the glomerular filtration rate. So we choose B as the correct answer there. Okay. Okay, so we can look at question 49. So question 49, they're saying which of the following is not one of the chaga fruits? Okay, which of the following is not one of the chaga fruits? So A, they're saying the nitrogenous base composition of DNA obtained from different species is not the same. Okay, B, they're saying the nitrogenous base composition of DNA obtained from different parts of the same species will be the same. Okay, so this one is part of the chaga fruits. This one is part of the chaga fruits. And then see, they're saying age, diet, or indeed any environmental factor does not affect the nitrogenous base composition. Okay, so yes, this is also one of the chaga fruits. And then D, they're saying DNA is double-stranded and the two strands of the DNA run antiparallel to each other. So this one, no, it's not a chaga fruits, but instead this was a finding, okay, for Watson and Crick. So in the Watson and Crick model, they discovered that DNA is double-stranded, okay, and those two strands of DNA, they run antiparallel to each other. Okay. Okay, so meaning this one here is not one of the chaga fruits. This one here was by Watson and Crick. Watson and Crick. So that would be the answer here. So you pick D because they said which one is not. So make sure you will be reading these questions carefully. Okay. The other one here, they're saying the only amino acid without a chiral carbon and therefore does not show any stereomylism is what? It's definitely glycogen. Okay, I mean glycine, not glycogen. Okay, so glycine it's attached to two identical hydrogen. Okay, so it lacks chirality or simply meaning the attachment to four different groups. Okay, so it does not have a chiral carbon. So the answer there is A. 49, they're saying which of the following is not a biomedical importance of amino acids? Okay, so let's read here. So A, the synthesis of biologically active amines like serotonin, histamine, and um, no, norepinephrine. Okay, norepinephrine. So this is a significant, okay, this is a significant importance of amino acid. It produces these um, um, hormones, these ones here. Okay, so this is significant. It's a monomer for monomer units for proteins. Yes, this is biologically important. Uh, 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 Biological importance of the amino acids. And then regulation of pH, yes, it's also important. And then here, constitution of biological membranes, no. Okay, so this one here is not about medical importance of amino acids. The ones that do this are what? Lipids or fats. They are the ones that are found in biological membranes. So the correct answer there is D, because they said it's not there. Okay, let's look at the last question in this section A. So section A, we're looking at the last question, which is 50. So they're saying um, the proximal histidine, okay, found on the alpha helix F is the point of attachment of the polypeptide to the heme group. This is important for preventing oxidation of iron atom, okay, in the heme group from the ferrous ion there to the ferric ion. What is the other role of this attachment of histidine, okay, F8 to the heme group? Okay, so here, before I say the answer, maybe I should show you a diagram. Okay, so the diagram for this heme, okay, it's something like this. Where's the diagram? So this is the diagram here. Okay, so you can see the heme group here. So in the middle there, we have these iron 2 plus ions. Okay, and then this iron 2 plus ions, it coordinates 6 bonds, of course. Okay, and it's the center of the porphyrin ring. Okay, so this is just a nice visualization, okay, of the oxygen that is being bonded to there. And then you also have this guy here. So now here I'm showing the proximal and the distal histidine. So the proximal histidine is called F8. The distal histidine, okay, is called E7. Okay, so this here is the iron 2 plus, like this one. And then you have these two oxygen that have been bonded there. You've seen these two oxygen here. Okay, and then this whole thing here, the red thing there is what we call the heme. Okay, so the question here was saying that what is the other role of the attachment of the histidine F8 to the heme group? Okay, so the other role is that it prevents oxygen, okay, from binding tightly to the heme group. Okay, so this proximal, this guy here, it prevents this oxygen from binding tightly to the what? To the heme group. Why? Because if it binds tightly to the heme group, then it won't be released to go and circulate. Okay, so the person will suffocate. So the proximal histidine, this one here, the F8, okay, it direct bounds. Okay, we can see it. It direct bounds here. This guy here, it direct bounds, okay, to the F2 plus, okay, in the heme. You can see the connection there, right? Okay, so this makes sure that it anchors the polypeptide to the porphyrin ring. This one is the porphyrin here, the heme. So this connection keeps, okay, this connection of this proximal histidine tightly bound to this F2 plus, okay, in this heme. This connection keeps, okay, the F2 plus slightly out of the porphyrin plane and ensures oxygen binds irreversibly, okay? And, or in short, I mean, it ensures this oxygen here bounds not too tightly, okay? So because the oxygen binds not too tightly, okay, then it would be released like that. But if the oxygen were to bound strongly, okay, to this heme here, then the hemoglobin wouldn't release it to the tissues, and then the person will suffocate, okay, due to lack of oxygen. So when we go back to the options now here, I'm just looking for the option that says the oxygen should prevent the binding tightly to the what? To the heme group. Okay, so we have that option on A. So the other option of this histidine F8 attachment to the heme group is that it prevents oxygen from binding tightly to the what? To the heme group. Okay, so why are these all other options wrong? Okay, for example, B, it prevents oxygen from binding perpendicular to the plane of the flat tetra pyro there. Okay, so this one is wrong because this prevents oxygen from binding perpendicular. Okay, this guy here, the histidine F8, it prevents oxygen from binding perpetulia. Okay, um that is um okay. B, they're saying prevents oxygen from binding perpendicular to the plane of the flat tetra. Okay, so I made a mistake. So this one here is the role of the distal. Okay, so it's the role of the distal. So the disto prevents oxygen from binding perpendicular to the plane of the flat tetrapyro there. Okay, so this one is the function of the disto. Okay, and then this histamine F8 is the same as the proximal. So the proximal is to prevent oxygen from binding tightly to the heme group, and then the distal is to prevent oxygen from binding perpendicular to the plane. Okay, and then C and D, these are incorrect. Why? Because histidine assists rather than prevents oxygen binding altogether. So you can see here they're saying prevents oxygen from binding from the heme group. No. Prevents oxygen from binding to histidine. No. So the correct answer there is A. So we are done with section A. Okay. As I said, in this video, we did question 33 to 50. And then question 1 to 32, the video is already there. Okay. So we're done with section A. And then for section B, okay, for section B, I'm going to do it in a separate video. Okay, I'm going to do it in a separate video.